To ensure that the information contained within the DNA does not become tainted, the information in the DNA is not directly converted into proteins, but first must be copied into RNA. The process in which DNA is converted into RNA is called transcription. The transcription of genetic information is the first step of gene expression, and its product is called a transcript. In eukaryotes, transcription occurs in the nucleus, while it occurs in the cytoplasm in prokaryotes since nucleus is absent. The enzyme that is responsible in this process is called RNA polymerase. So, let's get to know what RNA polymerase is, its structure, functions, and types. The RNA polymerase enzyme is a large complex made up of multiple subunits. The prokaryotic form of RNA polymerase has 4 subunits capable of transcribing all types of RNA. In eukaryotes, these enzymes have 8 or more subunits that facilitates the attachment and processing of DNA throughout transcription. There are 3 stages of transcription: the initiation, elongation, and termination. The 3 stages of transcription involve various functions of RNA polymerase that result in the synthesis of RNA. Initiation begins when the RNA polymerase wraps around the promoter region of DNA. The promoter is a DNA sequence that guides the RNA polymerase on where to bind upstream of a gene. While prokaryotic RNA polymerase can directly bind to the DNA promoter sequences, eukaryotic forms require the assistance of transcription factors for initial binding. Once RNA polymerase successfully binds DNA at the targeted promoter region, the enzyme can continue with the second stage of transcription. Elongation commences when RNA polymerase unwinds double-stranded DNA into 2 single strands. These DNA strands are used as genetic template for RNA synthesis. As the DNA template strand moves through the RNA polymerase, it builds an RNA strand that is complementary to the transcribed DNA strand. Termination is the final step of transcription. Once RNA polymerase encounters a terminatory sequence or signal, it stops adding complementary nucleotides to the RNA strand. This is followed by the release of RNA transcript, which marks the end of transcription for the template of DNA. While prokaryotes like bacteria have one RNA polymerase that transcribes all types of RNA, eukaryotes like plants and mammals can have numerous forms of RNA polymerase. RNA polymerase 1 is responsible for synthesizing most ribosomal RNA transcripts. These transcripts are produced within the nucleolus, a region within the nucleus where ribosomes are assembled. The availability of RNA molecules produced by RNA polymerase can impact essential functions of cell biology since these transcripts are directly involved with the production of ribosomes. RNA polymerase 2 transcribes protein-coding genes into messenger RNA. This 12 subunit enzyme works as a complex that directly influences gene expression through its production of pre-mRNA transcript. Once the pre-mRNAs are released by RNA polymerase 2 within nucleus, biochemical modifications prepare these transcripts for translation. RNA polymerase 2 also produces microRNA molecules. These non-coding transcripts can mediate gene expression and the activity of mRNAs after transcription. RNA polymerase 3 transcribes rRNA genes into small RNAs like transfer RNA and 5S rRNA. These smaller RNA transcripts play a role in normal cell function throughout the nucleus and cytoplasm. The last 2 types of RNA polymerase are exclusively found in plants. RNA polymerase 4 and 5 are transcription enzymes that evolve as specialized forms of RNA polymerase 2. Both enzymes produce small interfering RNA transcripts, which play a role in the silencing of plant genes. So, let's do a recap. Transcription. Transcription is the process in which DNA strand is converted into RNA strand. Transcript. A transcript is the product of transcription. There are 3 stages of transcription, which include the initiation, elongation, and termination. And there are 5 types of RNA polymerase, which include the RNA polymerase 1, 2, 3, and RNA polymerase 4 and 5, which are exclusively found in plants. And that's all for today's video. I hope you learned something new today and have a great day ahead.